Welcome to this exploration of microfluidic chips and how they're revolutionizing medical diagnostics. Today we'll understand how these tiny devices enable rapid testing at the point of care, from immunoassays to nucleic acid tests. Let's start with the fundamentals. What exactly is a microfluidic chip? A microfluidic chip is a miniaturized device that manipulates extremely small volumes of fluids, typically in the microliter to picoliter range. These chips contain channels that are typically 10 to 500 micrometers wide. To put this in perspective, a human hair is about 100 micrometers in diameter. Here's a simplified diagram of a microfluidic chip. You can see the inlet where sample enters, the network of tiny channels, and the outlet. The channels form a network that can mix, separate, or analyze the fluid. Now you might ask, why does making things smaller actually improve diagnostics? First, smaller volumes mean you need much less sample from the patient. Instead of milliliters of blood, you might only need a single drop. Second, reactions happen much faster, because molecules don't have to travel far. Diffusion distances are tiny, so mixing and binding occur rapidly. Third, you use far less reagent, which reduces costs dramatically and produces minimal waste. Let me give you a back-of-the-envelope calculation to show you the power of miniaturization. A traditional lab test might require 1 milliliter of sample. A microfluidic test might only need 10 microliters, which is 100 times less. That's a 100-fold reduction in sample volume. For expensive or rare samples, this is transformative. Now let's understand the physics that makes microfluidics work. The key phenomenon is capillarity. At the microscale, surface forces become much more important than volume forces like gravity. This is because surface area scales as length squared, while volume scales as length cubed. Surface area is proportional to length squared. Volume is proportional to length cubed. The ratio of surface to volume goes as 1 over length, so as you make things smaller, surface effects become dominant. This means capillary action can pull fluid through channels without needing external pumps. The liquid naturally flows due to surface tension. Here's capillary action in a narrow tube. The liquid rises higher than the surrounding level because surface tension pulls it upward. In microfluidic channels, this effect is very strong. We can calculate the height of capillary rise using the Jurin equation. Height h equals 2 times gamma times cosine theta divided by RHO-GR, where gamma is surface tension, theta is contact angle, rho is density, g is gravity, and r is radius. For water in a 100 micrometer channel, let me calculate the capillary rise. Surface tension of water is 0.072 newtons per meter. Assume perfect wetting, so theta is 0 degrees, and radius is 50 micrometers. Plugging in the numbers, we get a capillary rise of about 29 centimeters. That's remarkable. The water can climb nearly 30 centimeters purely from surface tension. Beyond continuous flow, there's another powerful approach called digital microfluidics, where we manipulate individual droplets. In digital microfluidics, individual droplets act as tiny isolated reactors. Each droplet can contain a different sample or reagent. These droplets can be precisely moved, split, or merged using electrical fields. This is called electrowetting. Here we see 2 droplets, one with sample and one with reagent. They're moved toward each other and merged to form a single mixed droplet where the reaction occurs. You might ask, why is droplet control so powerful for diagnostics? Because each droplet is essentially a separate experiment. You can run many tests in parallel on the same chip, dramatically increasing throughput. There's no cross-contamination between droplets because they're physically separated. This is crucial for accuracy. Now let's explore how immunoassays work on microfluidic platforms. Immunoassays detect specific proteins or antibodies. The principle is simple. Antibodies bind very specifically to their target antigens, like a lock and key. Here's the chip surface coated with capture antibodies shown in red. When sample flows over, target antigens in blue bind to these antibodies. A typical microfluidic immunoassay has 3 main steps. Step 1. Sample flows over the surface where capture antibodies are immobilized. Step 2. Target antigens bind to the capture antibodies. Unbound material washes away. Step 3. Detection antibodies with fluorescent or enzymatic labels bind to the captured antigens, creating a sandwich. The time advantage is enormous. Traditional ELISA assays take 3 to 5 hours. Microfluidic versions can complete in 10 to 30 minutes. This is because diffusion time scales as length squared over diffusion coefficient. If the distance is 1 millimeter and diffusion coefficient is 10 to the minus 10 meters squared per second, the time is about 10,000 seconds, nearly 3 hours. But if we reduce the distance to 100 micrometers, the time drops to about 100 seconds, less than 2 minutes. That's a 100-fold speedup. Let me give you a concrete example. Detecting cardiac biomarkers like troponin for heart attack diagnosis. Troponin is a protein released when heart muscle is damaged. Elevated levels indicate a heart attack. Traditional lab tests require sending blood to a central lab and waiting 3 to 6 hours for results. A microfluidic point-of-care device can deliver results in 15 minutes right at the patient's bedside. This speed can be life-saving. These devices can detect troponin at concentrations down to picograms per milliliter, which is sensitive enough for early detection. Now let's move to nucleic acid testing, which detects DNA or RNA. This is crucial for infectious disease diagnosis. The challenge is that DNA and RNA often exist in very low concentrations in patient samples. The solution is amplification using polymerase chain reaction or PCR, which makes millions of copies of the target sequence. Microfluidic PCR has several major advantages over conventional PCR. First, thermal cycling is much faster because the chip has very low thermal mass. It heats and cools rapidly. Thermal diffusion timescales as length squared over thermal diffusivity alpha. For a conventional PCR tube with 5 mm dimensions, thermal equilibration takes about 25 seconds per cycle. For a microfluidic chamber with 200 micrometer dimensions, thermal equilibration takes less than 1 second. This dramatically speeds up the entire PCR process. PCR requires 3 temperature steps per cycle: denaturation at 95 degrees, annealing at 55 to 65 degrees, and extension at 72 degrees Celsius. Here's the temperature cycling scheme. Sample flows through 3 temperature zones: 95 degrees to denature, 60 degrees to anneal primers, and 72 degrees to extend. Then it cycles back for the next round. Typical PCR needs 30 to 40 cycles to amplify DNA to detectable levels. The amplification factor is 2 to the power n, where n is the number of cycles. After 30 cycles, you get about 1 billion-fold amplification. This turns a few molecules into a detectable signal. Conventional PCR takes 2 to 3 hours for 40 cycles. Microfluidic PCR can complete the same process in 20 to 30 minutes due to rapid thermal cycling. A critical real-world application is COVID-19 testing, which detects viral RNA. Standard laboratory reverse transcription PCR takes 4 to 6 hours, including sample preparation and thermal cycling. Microfluidic rapid tests can deliver results in under 30 minutes, enabling immediate clinical decisions. Advanced systems integrate everything: sample lysis, nucleic acid extraction, amplification, and detection, all on a single chip. Now let's discuss the detection methods used in these microfluidic systems. The most common method is optical detection using fluorescence or absorbance. Fluorescent labels emit light when excited. Here, fluorescent molecules in green are excited by blue light from above. They emit green fluorescence that's detected from below. The intensity correlates with the amount of target present. Another approach is electrochemical detection, which measures electrical current or potential changes when reactions occur. The advantage is that you don't need bulky optical equipment. Electrodes can be integrated directly into the chip. An exciting development is smartphone-based detection, where your phone's camera acts as the detector. Modern microfluidic detectors can be incredibly sensitive, sometimes detecting single molecules using techniques like digital PCR. Digital PCR works by partitioning the sample into thousands of tiny compartments. Each compartment either contains target molecules or doesn't. By counting how many compartments are positive, you get absolute quantification without needing a standard curve. Now let's discuss some practical considerations and challenges that engineers face. One challenge is sample preparation. Blood, saliva, or tissue samples need to be processed, cells lysed, proteins removed, nucleic acids purified. Integrating multiple steps on one chip is technically complex. You need valves, mixers, temperature control, and detection all working together. Cost is critical for point-of-care devices. They must be manufactured at scale affordably, often using polymer molding or 3D printing. Regulatory approval is rigorous. These devices must demonstrate clinical accuracy, reproducibility, and safety. You might ask, if microfluidic diagnostics are so powerful, why aren't they everywhere yet? The answer is that significant technical and commercial challenges remain. Integration is difficult, manufacturing at low cost is hard, and convincing healthcare systems to adopt new technologies takes time. But the field is advancing rapidly. More and more point-of-care tests are reaching the market every year. Let me give you a quantitative comparison between traditional lab testing and microfluidic point-of-care testing. For a traditional lab test, sample volume is 1 to 5 milliliters. Time to result is 2 to 24 hours. Cost per test is $20 to $200, and it requires trained technicians and centralized equipment. For microfluidic point-of-care, sample volume is 10 to 100 microliters. Time to result is 10 to 60 minutes. Cost per test can be $5 to $50, and it requires minimal training with portable equipment. The impact on healthcare access is profound, especially in resource-limited settings where centralized labs don't exist. Looking to the future, where is this field heading? One direction is multiplexing, testing for many disease markers or pathogens simultaneously on a single chip. Another is wearable devices that continuously monitor biomarkers, providing real-time health data. Integration with artificial intelligence will enable automated interpretation of complex patterns and assist clinicians. Organ-on-a-chip systems are being developed that mimic human organ function, revolutionizing drug testing and personalized medicine. An interesting question: what are the fundamental limits of miniaturization in diagnostics? The answer is that detecting single molecules represents nearly the fundamental limit. You can't detect what isn't there. Consider this. If you have one molecule in 10 microliters, the concentration is 1 divided by 10 times 10 to the minus 6 liters times Avogadro's number, which is about 1.7 times 10 to the minus 19 molar. That's extraordinarily dilute. Detecting at this level requires sophisticated optics and labeling strategies, but microfluidic systems are approaching these limits. Let me do one more back-of-envelope calculation to show the economic impact of miniaturization. Consider an ELISA immunoassay for detecting a cancer biomarker. A traditional ELISA uses about 100 microliters of reagent per well. A microfluidic version uses about 1 microliter, a 100-fold reduction. If the detection antibody costs $500 per milligram, which is typical for specialized reagents, the reagent cost per ELISA test is 100 microliters times 0.01 milligrams per milliliter times $500, which is 50 cents per test. The microfluidic test costs only 1 microliter times the same concentration times $500, which is 0.5 cents per test. For 1,000 tests, you save $500 minus $5, which is $495 in reagent costs alone. Scale this to millions of tests performed globally, and the savings become enormous, making diagnostics more accessible. Let's look at a real example you're probably familiar with. Lateral flow tests, like pregnancy tests or COVID rapid tests. These are actually simple microfluidic devices that use capillary action to move sample along a strip. Here's the structure. Sample is applied to the red pad on the left. Capillary action pulls it across the strip. At the test line, if target is present, a colored band appears. The control line shows the test worked correctly. These tests require no external power or equipment. They're completely self-contained and user-friendly. They can be manufactured for under $1 each, making them accessible globally. The limitation is lower sensitivity than laboratory tests. They're designed for point-of-care screening, not definitive diagnosis. Let me show you a calculation about detection sensitivity. Question: how many molecules are needed for visual detection on a lateral flow test? The human eye can detect about 10 to the 12 fluorophore molecules per square millimeter as a visible colored line. A test line area is about 1 square millimeter. Sample volume is about 100 microliters. The detection limit is roughly 10 to the 12 divided by volume times Avogadro's number, which is about 1.7 times 10 to the minus 8 molar, or 17 nanomolar. This sensitivity is adequate for many applications like pregnancy tests, where hormone levels are relatively high. But for ultra-sensitive detection of rare biomarkers, you need amplification strategies or better labeling approaches. Let me summarize the key takeaways from this exploration of microfluidic diagnostics. First, miniaturization enables dramatically faster reactions and requires much lower sample volumes. Second, at the microscale, surface forces dominate, enabling capillary-driven flow without external pumps. Third, immunoassays and PCR that traditionally take hours can be completed in minutes. Fourth, point-of-care testing dramatically improves healthcare access and speed of diagnoses, especially critical in emergencies and remote areas. Fifth, significant challenges remain in integration, affordable manufacturing, and regulatory approval. Finally, the field is rapidly advancing toward personalized medicine, continuous monitoring, and even organ-on-chip systems. The future of diagnostics is undoubtedly moving toward miniaturization. Imagine a world where disease detection is nearly instantaneous and universally accessible, where you can monitor your health continuously, and where treatments are tailored to your individual molecular profile. Microfluidic technology is making this vision a reality, one tiny channel at a time.